Hello, my name is Duane Johnson. It is August 10th, 2018. This is part two of my lab video assignment, environmental influences that influence microbial growth or bacterial growth. This is pH testing and salt tolerance. So we're going to begin. This is the pH testing portion. So we're going to be testing active cultures incubated in nutrient broths at pHs of 5, 7, and 9. It's going to be testing the Saccharomyces cerevisiae as well as the Staphylococcus epidermidis. 5, they're all labeled. The 5 is acidic, 7 neutral, and the 9 is basic. So we're going to get started. So we're going to start with the more acidic nutrient broth. So we're going to get them to the pHs first and then add the active culture later. So to begin, we're going to have to flame each tip, sterilizing each one. And then this is our hydrochloric acid, HCl. We're going to be adding 1 drop to each of the 5s. I'm going to bring them down to a pH of about 5, or exactly 5. So 1 drop each. That would be it. Now we're going to start, and then we're going to go to the 9s, the more basic. We're not going to do anything with the 7s because that's neutral. We're going to we're going to assume that the nutrient broths are about neutral because we're not going to add anything to them. So same steps, same procedure. Now this experiment's going to help us to understand how bacterial growth is influenced in various levels of pHs, or various pH levels. Acidic, neutral, and basic. And this is our sodium hydroxide, NaOH, 1 drop each as well. 1 and 1. So we're going to be inoculating first the Staphylococcus epidermidis. So before we start that, I'm going to just uncap all of these, make this a bit easier. And then we're also going to just reflame everything. Starting with our active culture. just flame all of these prior to the inoculation. Do that over again. Our sterilized pipette. So this requires 2 drops per active culture in each test tube. One, two. 1, 2. 1, 2. Goes back in, and in the bleach bucket it goes. Re-sterilize everything and recap. Alright, now we're going to move on to our Saccharomyces cerevisiae 5, 7, and 9. Just go ahead and uncap, sterilize, uncap, sterilize. And uncap, sterilize for 3. So the required incubation time for these is recommended 24 to 48 hours. And if nothing has occurred or is very apparent, you can do an additional 24. So here's our active culture. Sterile pipette. And 2 drops each again. One, 2. 1, 2. 1, 2. Now, I have forgotten to put the pH, the sodium hydroxide and hydrochloric acid in. So hopefully this little misstep doesn't affect it too much. So we'll go to our sodium hydroxide. It's going to go in the, that's the basic pH. 1 drop. And then the hydrochloric acid, pH 5, 1 drop. So hopefully if going out of the order of the steps, it won't affect the results too heavily, if at all. But I guess we'll find out with our experiment. And now into the incubation area they go for 24 to 48 hours, if not longer, another 24. And that's it. Thank you.